Hello, welcome back, everyone. So, what I want to do today is going through a specific type of studying. So, I have categorized all the things that I want to study about in one bout of session. So, we have general chemistry, organic chemistry, biochemistry, cars, one cars passage, some biology cards, flashcards, physics, and psych soc. So what we do here is that we have 7 categories, and I want to study till 2 p.m., which gives me an hour and 15 minutes. That would be 60 plus 15 minutes, that is 75 minutes. So if we divide 75 by 7, we have about 10 minutes for each category. So let's do it. And we set a timer for 10 minutes each time. And we can start. Let's change. Cool. Let's start it now. So we start with general chemistry. So for general chemistry, I'd like to start from the beginning and tackle the first chapters quickly. So, okay, I've gone over chapter 1 a lot, and I've even done the flashcards, so we can go to the second chapter. So, the second chapter is pretty easy peasy. It's about the periodic table, the alkali metals, stuff like that, the Zeph energy, all of them are increasing when you go to the right of the table, except the atomic radius, right? Yes. The atomic size decreases when you go to the right of the table, and also it decreases when you go to the top of the table. So, all the other attributes to the periodic table, such as uh, ionization energy, electronegativity, this is electron affinity, which is the only one that is 0, and also Zeph, the pull between the nucleus and the valence electrons, is unchanged when you go up and down in the periodic table. So you gotta know what is the difference between going up and down and horizontally across the periodic table. Going up and down is basically adding more shells, right? You go from the first shell to the second shell to the third shell to the fourth row, fourth shell. And when you're going to the right of the periodic table, you are increasing your valence electrons. So it is very hard to remove the seventh electron from the valence electron, because based on the octet rule, they all want to full fill their valence electrons. So they want to have 8 electrons in the valence orbital. And all this to say that across the periodic table, the electron affinity so goes up, increases when you go to the right. When you have your 7th electron in the valence electron. And there are some also subtleties that you need to know. For example, in the transition metals, when you have half of the d orbital, that is more stabilized, so that's more preferable, having half valence full rather than having like putting the 4s subshell in priority. So instead of having 4s2 3D like 4, we'll have 3D5 and 4s1 as far as I know, but we can definitely come back to this later. So that is for the second chapter, and there are common electronegativities. So we know that hydrogen is less electronegative than carbon, and carbon less than nitrogen, and nitrogen less than oxygen. And fluorine is the most has the most electronegativity, because when it gains the electron, the delta H of the reaction is less than 0 when gaining the electron, but the electron affinity is reported as positive value. And the force, it's here. The force the atom exerts on an electron in a bond is so the force between the atom and the electron in the bond is electronegativity. So there should be a bond, and the force that the atom exerts on the electron is electronegativity. Of the noble gases, only kratom and xenon have an electronegativity, Kr and Xe. So the only trend in the opposite direction is atomic size, which is cations less than neutral and anions being the most. Anions are negative and cations are positive, just like Na plus, K plus. But anions like Cl minus, F minus, O2 minus, right, P3 minus, stuff like that. So on the third chapter, we have covalent bonds. So covalent bonds are, what are they? It's based on the electronegativity, and when the two elements have similar electronegativity, they share electrons between them. So covalence is sharing of the electron, similar electronegativity. Bond order, it refers to whether a covalent bond is a single, double, or triple bond. As bond order increases, the bond strength increases, the bond energy goes up, and the bond length decreases. So when the bond energy is going up, does it mean that it's more stable? Based on the fact that the strength has increased, yes, it means it's more stable, right? So the non-polar bonds have an difference of electronegativity, delta En of less than 0.5. The polar bonds have electronegativity between 0.5 and 1.7, and the coordinate covalent bonds are single. It's when a single atom provides both bonding electrons, most often found in Lewis acid base chemistry. Single atom provides both bonding electrons. That's a coordinate covalent bond. Single atom. This is mostly in acid base, Lewis acid base. So why Lewis? Why not Bronsted-Lowry? What's the difference? So the Lewis is the lone pairs, right? So it's about donating the pair of electrons. Where do we have it? Let's see if we have acid base here. The gas phase solutions. Here we go. Lewis base is about the pair, you see? So Bronsted-Lowry is about donating and accepting hydrogen plus, the protons. It's the same definition as Arrhenius acid, donating, producing protons. But Lewis acid is accepting electron pairs. It's based on the electron pairs. So that's why we mentioned Lewis acid base for coordinate covalent bonds, right? There's also polyvalence, stuff like that. But we'll come back to the acid-base chapter very soon. This is a warp speed method of studying we're trying to do. So we're almost out of time. We have only a minute left. So let's finish chapter 3. So the hydrogen bond is OH, NH, FH, dipole, dipole. The strength increase is the most in hydrogen, then dipole, dipole, and then London dispersion. These are van der Waals forces and is a general term that includes dipole, dipole, and London dispersion forces. So van der Waals is these two together. The H bond acceptor is oxygen, and the H bond donor is nitrogen because oxygen is more electronegative. So it accepts the bond, so the electrons. And we have sigma and pi bonds, 1 sigma. This is easy. The formal charge. So this is the electronegativity. So more than 1.7 is ionic. More than between 1.7 and 0.5 is polar covalent, and non-polar is between 0 and 0.5. And ionic bonds are formed via the transfer of one or more electrons from an element with a relatively low ionization energy. So we'll come back to this later. So now we set the next timer. We have 0.7 minutes left we can dedicate to each. So here we do GC 2/3. Next action item AI. Finish GC 3. Now we go to organic chemistry. Told you this is warp speed. Just want to go quickly over everything. So we have done some sort of OC1, and I've done also the isomers. And let's actually go to bonding since it's related to just what we studied. We can go quickly over 4 as well. However, I think I've done this 3. Yes, I've done this before. So we can just do flashcards on it as well as 4. So let's do alcohols. Sure, why not? So alcohols have the general form ROH and are formed with the suffix ol. If they're not the highest priority, they are given the prefix hydroxy. Benzene ring with OH groups attached is phenols, named for the relative position of the OH group. So ortho is 1 and 2, meta is 1 and 3, and para is, so ortho, meta, and para. How do we memorize this? We need a mnemonics for this. O is 1, elemental P, M, P. Based on the alphabetic order, we can just L, M, N, O, P, O. So it's M, O, P. Never mind. You just need to know this is the one, the first one. Let's write it down here. Ortho is first one. And M and P in alphabetic order. Though we don't want to confuse this later. Like in the exam, you're thinking, oh, it was all in alphabetic order. So L, M, so M is 1 and 2, O is 1 and 3, and then P is 1 and 4. We don't want to do that. We don't want to make that mistake, right? Alcohols can hydrogen bond, raising their boiling and melting points. Sure. We know that hydrogen bonds are stronger, so it increases boiling and melting points. Phenols are more acidic than other alcohols because the aromatic ring can delocalize the charge of the conjugate base. So phenols are more acidic than other alcohols. because the aromatic ring can delocalize the charge of the conjugate base. So the charge of the conjugate base, like this, is delocalized, so they're more acidic. Being more acidic means that you have more hydrogen plus, right? You have that H. You're a proton acceptor and an electron giver, donor. Electron donating groups like alkyl groups decrease acidity because they destabilize negative charges. EWG, such as EN atoms, electronegative atoms, EWG, Van der Waals group, and aromatic rings increase acidity because they stabilize negative charges. So alkyl, electron donating, decrease acidity because they destabilize negative charges. And aromatic rings, EN atoms, increase acidity, like oxygen, stuff like that, right? Because they stabilize negative charges. It's about stabilization. So phenols are more acidic because they delocalize, they stabilize the charge of the conjugate base. Phenyl equals benzene ring Cr. The reactions of phenols, quinones, they are synthesized through oxidation of phenols. So if you oxidize phenols, you get quinones. And quinones are resonance stabilized electrophiles. They're resonance stabilized electrophiles. Vitamin K1, phylloquinone, and vitamin K2, the menaquinones, are examples of biochemically relevant quinones. So vitamin K is a good example, K1 and K2, of quinones, right? And this is the structure. Two double bond O's with the ring in between, and the double bonds are stabilized here. And they're electrophiles, so they want electrons, right? These oxygens, they want electrons. They are willing to accept the hydrogen plus. Hydrogen basically. Hydroxyquinones are produced by oxidation of quinones. So if you one level of oxidation of phenols, you get quinones, and another level of oxidation of quinones, you get hydroxyquinones. So adding a variable number of hydroxyl groups. So you add a variable number of hydroxyl groups. Oxidation is the loss of electrons. So when you introduce a hydroxyl group to this, you're getting an oxygen, so it's oxidation. Electrophiles, so if this is an electrophile, it wants electrons, so it gets the oxygen that has electrons. Ubiquinone, also called coenzyme Q, it's an oxidative phosphorylation, right? Another biologically active quinone that acts as an electron acceptor in complexes 1, 2, and 3 of the ETC. It is reduced to ubiquinol. Okay. 1, 2, and 3. Electron acceptor, electrophile. It loves electrons. Cool. We have almost a minute left. Reactions of alcohols, primary alcohols, they can be oxidized to aldehydes only by pyridinium, PCC, chlorochromates, PCC. So you can only oxidize primary alcohols to aldehydes only by PCC. They will be oxidized all the way to carboxylic acids by any stronger oxidizing agents. Any stronger oxidizing agent, carboxylic acid. Secondary alcohols can be oxidized to ketones by any common oxidizing agent. It doesn't matter. So alcohols can be converted to mesylates or tosylates to make them better living groups for nucleophilic substitution reaction. So mesylate and tosylate, better living groups, LGs, for nucleophilic SN reactions. Mesylates contain the functional group SO3CH3, and tosylates SO3CH, like more carbonate is longer. Tosy is T is more in M. M is this, T is that. Aldehydes or ketones can be protected by converting them into acetals or ketals. So protected. So acetals is a primary carbon with two OR groups and an hydrogen atom. Ketal is a secondary carbon with two OR groups and acetal, ketal, deprotection, the process of converting an acetal ketal back to a carbonyl or catalytic acid. Carbonyl by catalytic acid. Okay, we can come back to all of these. These need way further studying, right? So we go to the next. So that was OC 5. Now we go to biochemistry. Anywhere that's not yellow, I'll just do it. So we have, let's do lipids and their function here. So we have structural lipids. Their characteristics: lipids are insoluble in water, yes, and soluble in nonpolar organic solvents. Nonpolar organic solvents. We need examples, right? Phospholipids, they are amphipathic and form the bilayer of membranes. So they contain a hydrophilic polar head and hydrophobic non-polar tails. The head is attached by a phosphodiester linkage and determines the function of the phospholipid. The head, which is it the non-polar or polar? It's the polar, right? It determines the function of the phospholipid. Saturation of fatty acid tails determines the fluidity of the membrane. So we have fluidity. Saturated fatty acids are less fluid. So saturated, which is the lack of double bonds, right? It's less fluid. That's why they're solid, right? Like animal fat compared to oils. Glycerophospholipids, that contain a glycerol backbone. So these have a glycerol backbone as well. We have sphingolipids. They contain a sphingosine backbone. Many but not all sphingolipids are also phospholipids with a phosphodiester bond. These are sphingophospholipids. We have sphingolipids and sphingophospholipids. the phosphodiester bond is what gives the phospholipid, right, linkage. The sphingomyelins, the major class of sphingophospholipid, and contain a phosphatidyl choline or phosphatidyl acid ethanolamine head group, part of the myelin sheath. So cool. We know these are in the myelin sheath. These sphingomyelins. Glycosphingolipids attach to sugar mides instead of a phosphate group. Cerebrosides have one sugar connected to sphingosine. Globosides have two or more. So cerebrosides, one sugar connected to sphingosine. These have glyco, right, attached to sugar mides instead of a phosphate group. Two or more is globosides. Gangliosides contain oligosaccharides with at least one terminal N-acetylneuroaminic acid, which is NANA. At least one terminal NANA. Vaxes, they contain long chain fatty acids esterified to long chain alcohols used as protection against evaporation in parasites in plants and animals. So long chain fatty acids esterified to long chain alcohols used as protection against evaporation in parasites. Use Vaxes. So this is a phospholipid, polar head, nonpolar tails. This is a phospholipid, fatty acid, glycerol. We use glycerol or sphingosine for the backbone. Phosphate alcohol. So these are the fatty acids, tails, and the alcohol, the phosphate is the head, and the glycerol is the backbone. They have cerebroside, a type of glycolipid. Any lipid linked to a sugar is a glycolipid. So any lipid that is linked to a sugar is a glycolipid. And this is the sugar unit, glucose, and then the fatty acid unit. And fatty acids like the amides, right? It's the carbonyl with the nitrogen attached. So it's not a carboxylic acid, but it's an amide, however you want to say that. So let's just continue quickly here. We have terpenes, terpenoids, steroids, steroid hormones, prostaglandins. So terpenes, adiferous, they have odor. Steroid precursors made from isoprene. One terpene unit, monoterpene, contains two isoprene units. Derived from terpenes, terpenoids are derived from terpenes via oxygenation or backbone rearrangement. Odorous characteristics. Oxygenation, not oxidation, or backbone rearrangement. Steroids contain 3 cyclohexane rings and 1 cyclopentane. 3 rings and 1 cyclopentane. Steroid hormones have high affinity receptors, work at low concentrations, and affect gene expression and metabolism. High affinity receptors work at low concentrations and affect gene expression and metabolism. We know that. Cholesterol is steroid important to membrane fluidity. and stability and serves as a precursor to many other molecules. The more cholesterol, the more fluid your membrane is, the more stable, right? Prostaglandins are autocrine and paracrine signaling molecules that regulate CAMP levels. That's for secondary messengers in the GPCR, right? Cycloaminopeptidase. Affects smooth muscle. Autocrine, paracrine. Autocrine is on itself. Paracrine is the juxtaposition. Affect smooth muscle contraction. Body temperature. Sleep wake cycle. Fever, pain. Prostaglandins. Vitamins A, D, E, and K. These are fat soluble. Vitamin A is keratin for vision. Vitamin D, cholecalciferol for bone formation. Vitamin E is called tocopherols. It's for antioxidants. Vitamin K, polyquinone, and menaquinone forms prothrombin, a clotting factor. This is a structure of steroid, 1-cyclopentane and 3-cyclohexanes. Vitamin K is for clotting factor 2. And energy storage, triacylglycerols, storage form of fatty acids, contain one glycerol attached to 3 fatty acids by ester bonds, very hydrophobic, so do not carry additional water weight. Triacylglycerols, storage form of fatty acids. So storage, one glycerol attached to 3 fatty, very hydrophobic, so do not carry additional weight. Additional water weight. Adipocytes, animal cells used specifically. 20 seconds left. Animal cells used specifically for storage of large triacylglycerol deposits. Free fatty acids, unesterified fatty acids that travel in bloodstream. Salts of free fatty acids are soaps. Saponification, the ester hydrolysis of triacylglycerol using a strong base. The ester hydrolysis of triacylglycerols using a strong base. Ester hydrolysis, like sodium or KOH. Micelle can dissolve a lipid-soluble molecule in its fatty acid core and washes away with water because of its shell of carboxylate head groups. Cool. We're good for now on the lipid chapter. It's a lot to learn in one bout, but we're gonna dive deeper into each concept later in the second try we do. So that was chapter 5, I think, for biochemistry. BC 5. Now the cool part, which is the cars. Okay, I'm probably going to do it silent. Or since I've been talking a lot, I might even do this one out loud, but we'll see. But if you want to do it in 10 minutes, we have to. I have to do it silent, so I'm sorry. Let me just set the timer. We can set the timer for this one here, actually. So we have 7 questions that would take us. Let's do this one, perhaps. Yes, this is 5 questions. We should do it in 8.5 minutes to be safe. Let's do it. Let me just zoom a bit. Yes. guess more like that. Not that they were all surrounded by ditches. This one was specifically in Cossack. This was specifically in the ditch, but they made it in such a way that it helped the invaders cross the ditch. The claim that these enclosures were not farm corals. Most intriguing interpretation. Excited. The answer to which informs the internality of the author's argument. How much did this is not about architecture, but it was religion to me. This is not about religion. It's about enclosed places, like for what did people believe happened to those sacrificed. It's not also important. Also, religion was discussed. The main idea is Neolithic enclosures were used for religious purposes. Oh. That's not important, right? Yeah, I should have chose that one. I'm pretty sure I got the rest right. Made a stupid mistake. We don't want to make stupid mistakes. That was... Wait. Where would be most challenged by the finding that they would often feast and sacrifice captives in their fortifications. Measurement of what the Neolithic enclosures were would be most challenged by finding that Neolithic armies would often feast and sacrifice captives in their fortifications. Oh, the period's farmers usually kept small herds of livestock. Why did I choose this one? Kept small herds of livestock. Oh, that's also not bad. Let's look at this one first. We need to identify the finding that would most challenge the author's explanation. The main idea is Neolithic enclosures were used for religious purposes. The author argues that they were not used for defense or farming. The problem I had was just I should have highlighted this as the main idea of the passage, right? The author argues that they were not used for defense or farming. A finding that contradicts this would provide evidence that the enclosures were used for one of these purposes instead. We can eliminate answers that are in line with the main idea or that go beyond the information in the passage. A. We might be drawn to this answer because of the discussion in paragraph 4. There, the author says that the enclosures were not used for farm animals because there was no phosphate on the ground. Also, the entrances, because there the author says the enclosures were not used for farm animals because there was no phosphate on the ground. Also, the entrances were too narrow. If it was found that farmers kept herds of livestock, we may think this changes the. While this answer says they kept livestock, it does not say that they kept livestock in the enclosures. Fuck. They could have kept livestock in another location. Also, even if they kept small herds of animals, each individual animal would still have not been able to fit through the entrance. The passage argument depends on the size of the animals, not the size of the herd. This answer contradicts. The main idea is the Neolithic enclosures were used for religious purposes. The fifth and sixth paragraphs mention that there is evidence of feasting and human sacrifice. Both of these activities were an important part of religious ceremonies. However, if Neolithic armies were the reason for feasting and sacrifice, it would contradict the main idea. Paragraph 2 states that the enclosures were not military installations because there are no indications of violence and destruction. But if they were used by armies for holding feasts, it would suggest that the enclosures did have a military purpose. It would contradict the claim that the enclosures were used for religious practices. Thus, this answer weakens the author's argument. Neolithic armies would often lead feasts and sacrifice in their fortifications. Okay. I apologize for that. I think I got the passage in like why I got those wrong. Poor guy got a 1 out of 5. 2 out of 5. It was relatively kind of hard for people. I think question 4 was easy, right? Yeah, it's in the passage. The author said, excited. You have to look for the words near the idea and size of the enclosure entrances. The claim that these enclosures are not farm quarters is based mainly on the size of the enclosure entrances. This one also got me. Should have chosen C. How important was religion to Neolithic peoples? That was the main idea, not how far were these enclosures from Neolithic settlements. I think I rushed it because I just wanted to do it in less than 8 minutes and 30 seconds. So lessons learned for this one. So let's continue with our progress. Did cars passage Neolithic enclosures entrances. Now let's do some biology flashcards. Okay, penetrance. This is a genetic terminology, and it's the proportion. So there is a difference between penetrance and expressivity. So penetrance is how much of the population is wild type now, instead of a mutant. Sorry, instead of wild type. And expressivity is the intensity, like the degree of variability that you see in the phenotype. The penetrance is just the proportion of the phenotype, proportion of individuals carrying a particular variant of a gene, allele, that also express an associated trait. So there is an associated trait with it, right? And that's when we have complete penetrance is all mutants. We can have with limited expressivity, and if it has expressivity, you have degrees of the dominance, the mutant type, but no wild type. But incomplete penetrance, you still have the wild type, and if the expressivity is also high, you get a degree of wild type to mutant. So the key apparently word here is that they also express an associated trait. So the proportion of those individuals that carry a particular variant of a gene also express an associated trait. Genetic leakage is when this is the interbreeding, right? This is the zebra horse, and not to be confused with genetic drift. Genetic drift is when basically the one, like some environmental incident, like a bird taking all the yellow flowers, drifting it into another place. So genetic leakage is just genes flowing from one species to another via hybrid offspring. So you get a hybrid car. Sorry, zebra horse. Genetic drift is when the composition of the gene pool changes as a result of chance. So like the wind, right? Or that was actually the founder effect. So genetic drift is just based on chance. When the composition of the gene pool is completely based on chance, not natural selection. This is the important concept. The founder effect is when the bird takes one species and there's the concept of inbreeding that happens here. Inbreeding. So is the loss of genetic variation. So genetic variation is lost. It occurs when a new population is established by a very small number of individuals. Mendel's law of independent assortment states that, so the independent assortment law is basically when the chances of one allele gene being expressed is not affected by another allele, and the exception to this is linked genes, and linked genes are an exception to independent law of independent assortment unless they go through recombination. So the alleles of two or more different genes get sorted into gametes independently. This is all about gametes, right? So the gametes are these different genes are sorted into gametes independently of one another. Their chances don't affect each other. It happens during meiosis, and it's during metaphase 1, right? When they are assorted independently of one another. Now, genotype is when one has, so it's the genotype is the variation of a gene, like an allele one has at a specific locus, at a given locus, combination of alleles, right? That's the genotype. And this is honestly a hard one for me, but we know genotype is just the BB, like heterozygous or homozygous genotype, right? So it's the variation of the alleles. One has a given locus. The locus is on the gene, right? A single twitch is when one muscle fiber is excited, right? Only one muscle fiber responds to a brief stimulus. Excitation, excited. Clonus is the rapid involuntary stretching and contraction, contraction and relaxation of a muscle, like the foot, going back and forth, rhythmic also. During muscle contraction, CA2 plus binds to troponin, causing a shift in tropomyosin, causing the attachment of the myosin head to the actin filaments, to the myosin binding sites on the actin filament. During muscle contraction, shortening of the sarcomere occurs as myosin heads bind to the exposed sites on actin, forming cross bridges and pulling the actin filament along the myosin filament. And pulling the actin filament across the thick filament, myosin filament. Muscles relax when acetylcholine is degraded by acetylcholinesterase, terminating the signal and allowing calcium 2 plus to return to the sarcoplasmic reticulum. ATP binds to the myosin head, allowing it to release from the thin filament, actin. In what creatine phosphate is what adds a phosphate group to ADP, forming ATP. In creatine phosphate pathway, creatine phosphate as a phosphate in muscles. Creatine phosphate is what adds a phosphate to ADP, forming ATP. Hypotonia, commonly known as floppy baby syndrome, is a state of low muscle tone. Muscle spindles are stretch receptors within the body of a muscle that do what? They sense stretches, right? Primarily detect changes in the length of the muscle. Changes in the length of the muscle, that's their primary function. If the muscle is shortened or stretched, it is detected by the intrafusal fibers of the muscle. Intrafusal fibers of the muscle. These are surrounded by the normal fibers known as extrafusal muscles. The corticospinal tract is for the movement of our trunk and limbs. Is a white matter motor pathway starting at the cerebral cortex, the terminus on lower motor neurons and interneurons in the spinal cord. The purpose is to move the trunks and limbs, controlling movements of the limbs and trunk. Corticobulbar is for our faces. An allele is a variant form of a gene located where? It's located on the genotype at the same position on the chromosome. It's located, so a variant of a gene located at the same position on the chromosome. Most genes exist in, so it's like on sister chromatids, right? Exist in many forms called alleles. An allele is any of the alternative forms of a gene that may occur at a specific locus. Our cells have 2 alleles for each gene, one from each parent. Incomplete dominance, like getting green from the blue and yellow alleles, is a form of intermediate heterozygous inheritance in which one allele for a specific trait is what and has to have 2 homozygous of different alleles to let this happen. Dominance is not completely expressed over its paired allele. For a specific trait is not completely expressed over its paired allele. You essentially make a third trait a new color, for example. Chromosomal, we have 3 seconds, so it's over for this. These flashcards take time, honestly. So we're done. I think we did like, let's see, 18 cards in 6.7 minutes. Not bad. 18 cards in 6.7 minutes. Or we can just do this. And now let's do physics. I haven't done physics for a long time. And I know most pre-meds hate it. I've been actually doing the physics chapters from the end, since it's kind of not related, and even better to do from the final chapters. So let's do chapter 10. Let's set our timer. So we have scientific notations. They improve the ease of calculation. These are about arithmetic and sig figs. Significant functions, I think. is usually helpful to convert a number to scientific notation. 3 to the 3 times 10 to the 3, 9 minus 9 times 10 to the 2 equals 3 times 10 to the 3 minus 0.9 times 10 to the 3 equals 2.1 times 10 to the 3. So 3, you just make this 0.9 times 10 to the 3, or you could make this 30 times 10 to the 2. So 30 minus 9 would give us 21 times 10 to the 2, which is 2.1 times 10 to the 3. Then you have 1.5 here, add exponents, just multiply them. For this here, subtract exponents. So you subtract 3 by minus 2 gives you minus 5, and this is 4. So when this goes on top to the top, like when you get when it's 10 to the 3 in the denominator, you get a 10 to the negative 3 in the numerator. And 2 times 10 to the negative 2 over, we have multiply exponents, so 10 to the 3, 8, 10 to the minus 6, negative 6. Then we have, this is radicals, so divide the exponent by 2, 9 gives you 3, and 10 to the 8 gives you 10 to the 4. Large mnemonic when moving the decimal within scientific notation. Left add, right subtract. So 40, I just do it in my head. 481.2 times 10 to the 7 is 4.8 times 10 to the 9. And this is 3.14 times 10, so 1, 2, 3, 10 to the 0, which is 1, so it's 3.14. Oh, my bad. So it's 3.14. You just want to double check, right? So it's 3.14 times 10 to the negative 6, because this is 1, 10 to the minus 1, 10 to the minus 2, 10 to the negative 3. Significant figures, sig figs, include all non-zero digits in any trailing zeros in a number with a decimal point. So a number with a decimal point, 3 point something. They include all non-zero digits in any trailing zeros in a number. Estimation, multiplication. If one number is rounded up, the other should be rounded down in proportion. Division, if one number is rounded up, the other should also be rounded up in proportion. Trigonometry, SOHCAHTOA, sinus, opposite over hypotenuse, cosine, adjacent over hypotenuse, and tangent, opposite over adjacent. So sine over cos. SOHCAHTOA, SUCTO, SUCKERTO. Common values. So the cosine for 0 degrees here, so cosine is adjacent over hypotenuse, which is 1, because your adjacent is 1 and your hypotenuse is 1, right? And the sine here is 1, so for 90 degrees and 270 degrees, or 3 pi 2. This is pi over 2. This is pi. And the cosine is negative 1 here. So for 30 degrees, the sine is half. For 60 degrees, the cosine is half. For cosine of 30 degrees is square root of 3 over 2. Adjacent over hypotenuse, square root of 3 over 2, 1 over 2. And for 90 degrees we had it. For 45 degrees it's for both is square root of 2 over 2, but for tangent it's 1. Tangent of 30 degrees is square root of 3 over 3, and for 60 degrees is square root of 3. For 90 degrees the tangent is undefined because you will get a 1 over 0, which is infinity. And for 180 degrees we have negative 1 over 0, 0 over negative 1, which is 0 basically. And we'll just get to the logs in a second. But here we have 45, 90 triangle, square root of 2x, xx. And for 30, 60 triangle, this is 2x, x, and square root of 3x for the angles. Estimating square roots. But honestly, for triangles, I think Pythagorean theorem is the best, since you can just get a second exponent of each angle, and the hypotenuse is equal to the hypotenuse to the 2 equals x to the 2 plus x to the 2. To calculate the square root of any number less than 400, you can approximate its value by determining which two perfect squares it falls between. For example, square root of 180 is between 13 and 14. So less than 400, you can approximate its value by determining which two perfect squares it falls between. So 180, so for 13, yeah, whatever. So square root of 4 times 9 times 5, 2, 3, 5, 13.2. So 2, 3, 6, square root of 5, 2.2, because square root of 4 is 2, square root of 9 is 3. So it's 2.2. And 2.2, so 6 times 2.2 is 13.2. The common square roots are these. 11 is 121, 12 is 144, 13 is 169, 14 is 196, 15 is 2225, 16 is 256, 17 is 289, 18 is 324, and 19 is 361. Log and natural log of a equals b, so 10 to the b is equal to a. Natural log of, so e to the b equals a, and e is 2.7. Log a of 1 equals 0 because 10 to the 0 equals 1. 10 to the what equals a. 10 to the a of, so 10 to the what equals a is based on a. So log of one number over its own number is, so a to what equals a is 1, right? Log a greater than 1 is positive, log a less than 1 is negative. And log a times b inside it is log a plus log b. Log a over b is log a minus log b. Log a to the b, you bring the b to the back, b log a. And log 1 over a is minus log a. So this one is estimating log, 7 seconds. Log a times 10 to the b, log a plus log 10 times 10 to the b, log a plus b, log, and log a times 10 to the b, b plus 0.a, and boom, tangent sine over cos. Good. So here we just had 10 to the something. It's b itself, log a plus b. So log a plus b is actually log a times 10 to the b. So b plus 0.a. Y 0.a. Anyway, we can add question marks here. So last but not least, this was chapter 10. Last but not least, we have psych-soc. Let's do last 10 minutes. So the psych-soc was here that I've been doing some Khan Academy videos recently as well. But let me just continue and finish with, let's actually do chapter 6. But I didn't do James Long theory. I did these on my own, but I'm starting to quickly forget these. We need to do these reminders for these chapters. Expectancy value. The amount of motivation for a task is based on the expectation of success and the value of that success. Opponent process. The names are hard to relate it to the concept sometimes. Like you can have so many. So here's the problem with psychology concepts. You can have so many different ideas for the concept, but there's only one definite explanation for it. And so you have to be able to put them into words and explain them fluently, descriptively, with details, and attach them to examples and interpret them and make your opinions based on the data. So, yeah. Our limbic system is for our instincts and mood. You can look at the diagram for it. Self-concept and identity. So the sum of ways we describe ourselves is self-concept. The sum of ways we describe ourselves. Identities, individual components of our self-concept related to the group to which we belong. Self-esteem, the closer our actual self is to our ideal self. Actual to ideal. And our ought self, who others want us to be, this increases our self-esteem. Self-efficacy. The degree to which we see ourselves as being capable of a given skill or situation. So efficacy is our efficiency, right? Our skill sets. So it's about skill or situation. Learned helplessness. Basically, I think it's just learning that you're helpless. A state of hopelessness that results from being unable to avoid repeated negative stimuli. Like you can't stop watching porn. Let's say you can't stop using heroin. You can't stop smoking cigarettes. So it's learned helplessness. Locus of control is internal and external. So we control our own success or failure. Outside factors have more control is external. Formation of identity. Freud, psychosexual stages of personality development based on tensions caused by the libido, failure at any stage leads to fixation, which causes personality disorder. Oh, 0 to 1 is oral, 1 to 3 is anal, 3 to 6 is phallic, 6 to puberty is latent, puberty to adult is genital. Not to, so OAPLG. Not to confuse it. We have, I think, 3 of these at least. One is the Freud's, 0 to 1, 1 to 3, 3 to 6, 6 to puberty, puberty to adult. The thing with the thing is we had moral development stages, I think. Oh, we have moral development stages for Kohlberg. That's the second one. For Erickson, it's the third one. We also had the sensory motor, pre-operational, concrete operational, formal operational. For the, what was the name of the scientist? I forgot, but we can go back to that. Let's just do it here. Is it for for Piaget? Yeah. So Piaget stages. It's concrete operational and formal operational. Yes. So this one was 0 to 2, 2 to 7, 7 to 11, 11 and plus. For Freud is 0 to 1, 1 to 3, 3 to 6, 3, 6 to puberty, puberty to adulthood. And this one gets tricky. Stages stem from conflicts throughout life. So these are about conflicts, right? And the Freud is about fixation. It causes personality disorder from the fixation at any stage. So 0 to 1 is oral, 1 to 3 is anal, the 3 to 6 is phallic, 6 to puberty is latent, and puberty to adulthood is genital. For me, it's just actually easy to memorize it as OAPLG. OAP was the open air pub at McGill University, where all these juniors and freshmen and everyone is just like chilling there and drinking, and yeah, so OAPLG is for this. And then we have the Erickson stages. So 0 to 1 is trust. So this is my sexy girl in red is stacking dick. This is plus 18 plus. This is very R-rated, but this is what we have to do to memorize these mnemonics. So my sexy girl. So let's go. My mistrust versus trust. Yes. Sexy. Is it my sexy? So the sexy part of the S would be I don't know, shame versus autonomy. Okay, shame, sexy, my shameful girl, sexy girl. Let's say my sexy, shameful, shameful girl. And the G would be shame versus autonomy, girl. G versus guilt versus initiative. Guilt, my sexy girl, guilty girl. Guilty girl, my sexy girl. In inferiority versus industry. Yes. red role confusion versus identity. In red, role plays. In red, role play is isolation versus intimacy. Is sucking, stacking, stagnation versus generativity. Dick is despair. Sucking dick desperately. And the ages go 0 to 1 again, just like Freud, and then 1 to 3, just like Freud, 3 to 6, just like Freud, 6 to 12, 12 to 20, 20 to 40, 40 to 65, 65 to death. So again, my is mistrust versus trust, sexy or shameful, shameful, right? My sexy girl in red. My sexy shameful girl is guilt versus pleasure, guilt versus initiative. Guilt in inferiority, industry versus industry. In red role play, role versus role confusion versus identity. My sexy girl in red is isolation versus intimacy, sucking, stacking, stagnation versus generativity, dick, despair versus hope, integrity, despair versus integrity. Kohlberg, you can realize why Biden, for example, lacks integrity. I'm joking. Or any other old ass dude. It's because you're 65 plus. You're either in despair or you have integrity. Then we have Kohlberg stages based on moral dilemmas, 6 stages in 3 phases. Example, Mr. Heinz dilemma. So Kohlberg stages based on moral dilemmas, 6 stages in 3 phases. These Kohlberg ones are tricky as fuck. I never get to know them. Like, we need to be able to solve questions based on these, right? So Vygotsky, we have zone of proximal development. The skills that a child has not yet mastered and require a more knowledgeable other to accomplish. Intimation and role taking, common ways children learn from others. Reference group, the group to which we compare ourselves. Imitation and role taking, common ways children learn from others. And comparing ourselves, reference group, post. So pre-conventional, conventional, and post-conventional is the moral stages of Kohlberg. Pre-conventional, so these are the phases, right? 3 phases, and there's 6 stages in these. I must, so 2 stages in each. I must share this toy because if I don't, then I will get in trouble. You need to drive slower because the law says so. It's more about law. And post-conventional, just because the law says to do it doesn't mean it is ethical. That's intense. Okay, we have the personality on the right side to dig into later. So just know that it is unconscious, preconscious is superego, and conscious is ego. Okay, we'll get back to this. So for now, we did a full round, which was till 2 p.m. planned. So one half of chapter 6 was also done here. Cool. I'll see you in the next one. So we do have action plan for us here. I mean, not February 14th, but for tonight, I have to go to work right now. But for tonight, we'll have the GC chapter 3, OC6, I think, BC6, I think, cars, another cars, yeah, and BB, and physics 9, and Psych Soj 7, I think, 6 and 7. We need to review them ASAP, so we need to do these ASAP and review them ASAP. Okay, cool. I'll see you back.